So hello everybody. It's my pleasure to introduce first Gabriel, who is going to talk about the functional annotation within the BovReg project. The floor is yours.
Hello everybody, good morning. As Carol have introduced, I'm Gabriel Costa. It was former postdoc at the lab working on the functional notation of the bovine genome for the BovReg, and now moved for a new position in INJA. But here showing all the results that we have on the BovReg consortium on the multidimensional or high-quality functional notation of the bovine genome. What is our contribution on the functional annotation? With a bit of context, we know that epigenomic maps are crucial to better understand gene activity. As Kristen mentioned before, there are two important initiatives in humans, the ENCODE project, and also followed by the Roadmap Epigenomic Consortium, trying to follow up of some of the elements described and annotated on ENCODE project. And basically, on these two projects, they have identified thousands of cis regulatory elements in humans. In humans, about 900,000, and in mouse, about 300,000. They have a huge catalog of cis regulatory elements in human and mouse, and was it useful, and how they use, and what's the perspectives and possibilities with this data. They see that by using the epigenome map that cis regulatory elements annotated, they could refine about 30,000 loci that were previously associated with complex traits and disease-associated traits. So they can use the epigenome map to really refine GWAS loci that they have for diseases in humans, for example. We know that the majority of the loci that we have from GWAS with complex traits in livestock animals are falling into non-coding regions devoid of functional annotation for the moment for a diverse catalog of tissues. But it's important to remember here and highlight that recently there is a big discussion on human field about the lack of diversity on epigenomic studies and the impact it could have to dissect the different diseases loci associated. This is trying to be addressed on the new phase of Roadmap and ENCODE project on the phase 4, but we wanted to highlight here the importance of taking diversity into account when building high-quality epigenomic maps. And how we contribute and how we did it on the BovReg consortium. We have collected samples for 6 different animals, which are two neonate animals about 2 weeks old from Belgium, male and female, 2 which are hosting animals, 2 animals from Canada, which are Kinsella composite breed. They are juvenile, about 7 months old, male and female, and 2 animals from Germany, which are Charolais crossed with German hosting F2, sexually mature adult animals more than 1 year old, male and female. From each one of the animals, we have collected about 24 different tissue types from each one of the animals. And with this 129 catalog of tissues, we have applied the different assays on the transcriptome atlas with mRNA, total RNA, small RNA, cage experiment, and also on the landscape of regulatory regions with ATAC-seq, DNA methylation on RRBS and WGBS, and ChIP-seq for 5 different antibodies on the whole catalog of tissues of BovReg. I also include here, just to mention, few cell lines, 5 cell lines that have characterized and applied the different assays. There will be a talk of Doreen Becker from FBN on the next section about some of the characterization and structural variation on these cell lines. But just to say that we also characterize the 5 cell lines widely used for the livestock community. All this data were mainly produced by the same lab as Kristen mentioned before, and we analyzed it using the standard pipelines that were produced with a huge contribution of CRG, handled by CRG, on the next full NFCO compliant pipelines to analyze the data and release the functional annotation. And what we see on the transcriptome atlas of BovReg, putting together mRNA and total RNA, we are able to annotate about 47,000 gene models in bovine. If we compare with the other species, we can see how much we have increased the number of genes annotated in bovine, and these genes are supported by about 300,000 transcripts, which we annotated from poly-A and total RNA assays. From these 300,000 transcripts, about 43,000 were already predicted with biotypes on ensemble. We could annotate protein code or CDS, start and stop codon, for about 47,000 transcripts from this total, and we have about 200,000 transcripts as non-coding transcripts annotated on BovReg. When we compare with the existing annotations of ensemble and NCBI, we could see here highlighted, let me see, is this one for the light? Yes. Here highlighted on the table, we have the comparison with ensemble and NCBI, and we could see that as a perfect match with the annotation, we only have about 14% or 12% when comparing to both annotations. Then we have median changes and differences comparing to the reference, and we also have about 10 or 6% of the transcripts which are completely novel. There's any kind of overlap from what we know from the existing annotations of ensemble and NCBI. One asset of the BovReg transcriptome atlas is that we are taking into account total RNA. Most of the other studies are conducted using mRNA, and here we are also doing total RNA for exactly the same samples, and we have increased the catalog of non-polyadenylated transcripts annotated in bovine. Here I just put an example. It was a huge work by FBN to annotate the long no-coding RNA catalog using the data from BovReg. And here's just an example of a long no-coding RNA annotated on bovine, which is highly expressed on total RNA, but no expression at all on mRNA. So it's something that we are adding with non-polyadenylated transcripts on the BovReg annotation. When we look at the genes with elevated expression on the different tissues, here I'm showing the elevated expression for each one of the group of the tissues, and each color represents one of the animal from BovReg for the 6 animals. We can see that in testes, we have an elevated number of genes expressed. We know, we expect that. When we compare the neonate testes with the small bar with the adult testes with the huge bar in blue, we can see that for the adult testes, we have a huge number of genes with elevated expression that we know that are mainly related with late spermatogenesis, so it's expected. We also have an elevated number of genes with high expression on tissues originated from the central nervous system, which also meets our expectation. But more interestingly, when we compare the tissue that were represented on the 6 animals, only taking into account the ones that are represented for the 6, and we look at the potentially novel transcripts, the neonate animals, 2 weeks old, are the ones adding more new annotation of new transcripts. We have plenty of isoforms that are on the neonate animals 2 weeks old that were not taken into account on the current annotation of ensemble and NCBI. We also did cage experiment. It was the partner from the UK. As you see here, Emily Clark and Mazdek Salavati. This paper was already published, and the data is available for the whole community. It's publicly available, and what we see basically from cage assay. As Costa showed before in this graph, comparing with different species, by doing these assays and refining the annotation of TSS, we could identify about 4,700 TSS, which are exclusive from both starhos. It's specifics for both starhos. And when we look at the genome features of this TSS, the refined annotation of TSS in bovine, comparing with ensemble annotation, we could see that a huge percentage of them map to intergenic regions. And they are probably supporting the annotation of the new transcripts on the transcriptome map of BovReg that we need to intersect to support the annotation that we have for the moment. So this shows us that we still have room to improve the transcriptome atlas and the annotation of the different isoforms in bovine. But we asked ourselves, and also with the help of our reviewers, do we have a way to validate some of these transcripts? Are they true, and they could be captured by a different methodology? And we decided to do Oxford nanopore and sequence long-read cDNA in order to be able to validate if we can capture the same isoforms doing a different method. We did an experiment with 6 samples, and after this, we have a huge coverage for each one of the samples with Oxford nanopore. We assemble the transcripts, and we compare for exactly the same samples from the annotation of BovReg. By doing so, we could see that about 60% of the transcripts were a perfect match between BovReg annotation using Illumina short read technology and Oxford nanopore with long-read cDNA sequencing. So we have a strong evidence that we have plenty of true new transcripts that we are adding on the annotation of bovine. Here you can see one example with an IGV print screen of a gene which expressed, and before it was annotated was 2 different genes and 2 different models or long non-coding RNAs. And now we have one unique isoform which was annotated by BovReg and also supported by Oxford nanopore long cDNA reads. So we decided to proceed, and we are now finishing or close to finishing a huge catalog with a new transcriptome map using Oxford nanopore long-read cDNA sequencing with more than 1 million long reads from each one of the 129 different samples of BovReg. It will be the first huge catalog with high depth of coverage for the whole samples, the vast catalog of tissues of BovReg. We also expanded the repertoire of microRNAs annotated. Currently on the database, we have about 1,000 microRNAs annotated. With BovReg, we could annotate 1,285, for which half of it were predicted as novel microRNAs, and the other half as no microRNAs or with at least 1 hit with the database for the different species. But interesting, when we look at this novel microRNAs annotated, we have a huge proportion of them mapping to a specific DNA transposon family, which is not active in bovine anymore, but they are mapping to this family of transposon in bovine. We don't know yet if there is a biological meaning or if it's a technical issue, also because of the size of the reads for microRNA assay, but it's an interesting thing that we need to take into account, considering that this DNA transposon family only accounts for 0.6% of the genome. So we need to take this information into account and try to understand why we have this enrichment for a specific family of DNA transposon. But when we look on the novel microRNAs, indeed, as expected, most of them were already annotated in bovine, but we do have few that were annotated in different species, but not in bovine. And when we look to them, we have here one example of a microRNA that was not annotated before in bovine. It was annotated in different species, including sheep, but not bovine. And here on the first graph, we have the level of expression normalized by CPM for the diverse catalog of tissues. We can see that the high expression is only detectable on the neonate animal 2 weeks old, on the 2 neonate animals. So it's a microRNA with expression specific to the time point of neonate that we now could capture and add to the annotation of microRNAs. This microRNAs is annotated within a well-known imprinted domain with plenty other microRNAs annotated, and we are adding the annotation of this one because now we are using neonate animals about 2 weeks old. On the landscape of regulatory regions of BovReg, we have established for the first time a huge catalog of ATAC-seq and ChIP-seq peaks across a diverse catalog of tissues, applying ATAC-seq, ChIP-seq for 5 different antibodies. Here you can see on the different graphs the number of peaks detected by each sample, and we can see that the number and the coverage of the genome that they cover, the percentage of the genome that they cover, is pretty consistent between the different samples, except for one mark, which the A3K27 3-methylated, which is associated with polycomb repress, and we know that produces very broad peaks, which are difficult to annotate and difficult to really define the breaks of the annotation of the peaks. It's the one that I put my face question in the results just to highlight that this one is the one that is less stable across the different tissues. But what we did with this huge catalog of samples, we integrated on the prediction of chromatin states to be able to have the different states annotated on the catalog of tissues, combining ataxic and ataxic in ChIP-seq on these samples. Here in blue and white, we have the probability of emission of the different marks on the same position, and we selected 14 different states. By doing the prediction of the states, we could annotate about 25% of the cattle genome, which are way more compared to what we have on the literature for the moment. This excluding the state number 14, which they state with no signal at all in any sample. But we decided also to have a look on the active enhancers and CTCF and the regions that are predicted as active enhancers to see if we have something interesting. And by looking so, we have identified instances of active enhancer which are tissue-specific. In this example, it's liver-specific, which could only be detected on neonate animal, 2 weeks old. And it could not be detected on juvenile and adult animal, showing that indeed, by adding different stages, diversity, we have new annotation of active enhancers. Here I show some IGV pictures with the ataxic peak corresponding to the enhancer region, only in neonate animals, only in liver, when we consider the catalog of BovReg, and with ChIP-seq data corroborating that it's an active enhancer on this position. We also decided to look of the promoter TSS annotation of the chromatin states. We have transcriptome atlas, and we have TSS on the transcriptome atlas, and now we have the chromatin states with TSS and promoters. To what extent they overlap? To what extent they agree on the annotation? By doing the overlap and checking, we could see that about 79% of the TSS annotated on the transcriptome atlas are a match with the TSS regions annotated on the chromatin states. So we have a good agreement between the different techniques, improving the annotation of regulatory regions in bovine. But we also compared our predictions with the most recent paper with ChIP-seq and ataxic annotation in bovine, the paper from the team of Feng in US, where they used different 8 tissues to do ataxic, ChIP-seq, and predict the chromatin states. Here, to do a fair comparison, I'm only taking into account exactly the same tissues that they used. So here, it's only 8 tissues. On the table I'm showing here, we have the list of tissue. On the first column is the number of active enhancers annotated on the paper using the data of the paper. And on the third column is the number of active enhancers that we recapture using BovReg annotation. And in red, we have the percentage of the representation of the recaptured enhancers compared to the paper of UC Davis' team. And we could see that we could recapture most of the active enhancers annotated from Feng US paper. But interestingly, here I'm showing 2 examples, one for liver and one from spleen. In the graph, we have each one of the 6 animals of BovReg. In blue is the number of active enhancers that we are adding to the annotation. And in orange is the number of active enhancers that are common with the paper. It's common with the data set from UC Davis. So we can see that we recapture the active enhancers, but we are also adding new ones with the diverse catalog of tissues of BovReg. We are now trying to decompose and identify the different tissue-specific and tissue-shared components. Here's just an example with a picture where the different colors represents the different components for A3K4 3-methylated, which represents promoter and TSS region. In gray, we have the ones that are shared between the different tissues, and on different colors, we have the ones that are shared between tissues from the same origin or either tissue-specific elements. We can see that we have some promoter regions that are shared on endocrine system, on digestive system, muscular system, and we also have some components that are invariant that are shared for all the tissues and so on. We are using the methodology, which the methodology also used on the recent paper on the taxi catalog of cattle, and we are using the same methodology to decompose some of the chromatin states that we have predicted on BovReg. On the genomic structure variants, as you remember from the presentation from Costa, we were working on the function notation and structure annotation of bovine genome on work package 2. We have done the annotation of mobile genetic elements identifying polymorphic insertions in bovine. To do so, there was a collaboration between our team, Liege and Tang, which will have the next talk, and Menu from Switzerland, from Huber Posch team, working on the whole genome sequencing of different breeds to identify and apply a dedicated pipeline to identify polymorphic insertions of mobile genetic elements in bovine. We did it, we identified polymorphic insertions, and we deployed about 700 probes on urogenomics array to be able to genotype different cattle populations with mobile genetic element probes. When we intersect the probes of mobile genetic elements with our annotation of regulatory regions, we have observed tissue-specific regulatory elements co-localized with mobile genetic elements. Here's one example where we have an enhancer which liver-specific co-localized with a polymorphic insertion of ERV21 family. We know from the literature, I mentioned here in yellow one of the recent papers on pangenome, but we know from the literature that transposable elements will be increasingly identified as driving forces on phenotypic diversity in cattle, and taking advantage of the knowledge generated on BovReg and what we know from the literature, we have moved forward trying to unravel the genetic mechanism behind the de novo mobilization rate in bovine using specific family ERV21, which will be the next talk by Liege and Tang. The paper is already accepted. Congratulations, Liege. And we move a little bit forward on the annotation of these mobile genetic elements. Just an example, we have also looking at the catalog of elements from BovReg, we have identified instances of polymorphic insertions affecting and creating new polyadenylation signal. Here we have an example of one gene. In pink, we have the mRNA reads. In gray, we have the whole genome sequencing for a wild-type animal and also for an heterozygous animal for insertion of an ERVK family, BTLTR. And we can see that for the animal that has the insertion, the reads from the mRNA go through and stop on the position of the insertion. We even have chimeric reads on this position, and we can see that this polymorphic insertion is creating a new polyadenylation signal. We also analyzed the CNVs. We also identified CNVs on the 6 animals of BovReg, applying a pipeline on the lab with the help of Dr. Yang Lin, and inspecting the catalog of CNVs identified on the 6 animals. We have detected one animal with carrier of a known deletion, a lethal deletion, the haplotype 5, when it's lethal when it's homozygous. And here, I'm sure, oops, this is very sensitive. Yeah, and here we have the IHV picture showing in gray the whole genome sequencing of the animal. We have the position of the deletion that I'm highlighting in red. This deletion encompasses one gene, which is totally deleted, and also encompasses the last exons and 3' TR of a gene nearby. When we look at the transcriptome map of BovReg, we could see that this deletion is not only deleting the gene encompassed on the deletion, but it's also creating an alternative 3' end for the other gene nearby. We see it from Illumina short reads on mRNA and total RNA, and we also validated by Oxford nanopore long read sequencing. So we have the creation of an alternative 3' end nearby the site of the deletion. And taking advantage of the data generated on BovReg, we also decided to move forward and have a look and revisit some of the imprinted regions that we know in bovine. This work is currently ongoing, but just to give an example, here we have the well-known region of IGF2R, and I'm showing a print screen from IGV with the different stone marks, CTCF, mRNA, and whole genome sequencing of the animal. And we could see that when we have a look on the polymorphism that we have on the different marks, the allele that we have on the active marks is different from the allele that we have on the repressive mark, which is what we expect, but it allows us to refine and to really look at the imprinted regions and revisit them and characterize them. On the other hand, we can also look at some of the polymorphism that we have on the exons and see if we have monoallelic expression on this position, which is the case as we are showing here. So we are now able to revisit some of these regions and characterize and annotate the alleles that we have on the different active and repressive marks on the same region. It's an ongoing work for which Liege is heavily involved too, currently to move forward on this annotation. As a perspective and take-home message, the articles are coming. Stay tuned. We are putting together the big functional notation of the BovReg project with transcriptome landscape and the landscape of regulatory regions. So stay tuned, it's coming, and soon will be released for the whole community. I would like to thank all the partners involved on this huge work of 4 years and a half to putting together all the level of data on the same animal, same lab, and analyze the data. And I hope I managed to be within the 30 minutes dedicated for the presentation. Thank you very much.
Question for Gabriel.
Thanks, Gabriel. So I'm Emily Clark from the Russell Institute. So for the annotation, the new BovReg annotation, I guess the ensemble annotation is a sort of highly curated output from the gene build pipeline. Beyond BovReg, can we do more to curate the annotation that we've built for BovReg? So can we look more at genes that might be merged? Or, well, you mentioned using the cage data to validate the TSS. I guess maybe we could do that as a group, but also maybe ask the wider community to look at the annotation.
Yes, I think it's one of the goals. We are trying to do this within BovReg, but I think it's as you said, it's a very nice proposition for the whole community to put together the effort. We have the indication that after we release the data within BovReg and publish the data, the paper, it will go through the pipeline of Ensemble to increase and improve the annotation of Ensemble and release the data for the whole community. EMBO EBI, already agrees with this, and they will go through with this data. So it is something that will be for the whole community, but also open for the different collaborations and to move forward on the integration.
Thank you.
Okay.
Christa Kühn. I have actually 2 questions. One question is, when we looked into the transcriptome map, quite frequently we see very large transcripts. To what extent could you confirm them with the ONT? Because this is really striking when you look at the comparison to the current ensemble annotation. That's one. And the other thing is, how would your plans be regarding the integration of the single nuclear data? Because that's something we are currently also worrying about. Often we use bulk RNA-seq, but we are aware that this is just a more or less stable summary of different cell types.
For the first question, we could not confirm the most of the very long transcripts that we assemble from Illumina reads, because in fact, most of them are a technical issue, depending on the level of expression of the two genes or long code nearby and they're combined. The example that I show here, it's one example where we have a long transcript, but it's a true one, but they are not the majority. For most of the ones that we have on that, and using mRNA and tRNA and putting together, the very long transcripts we could not validate by Oxford Nanopore. This is true. It's more a technical issue that we still need to think as a community how to solve this issue, especially when using string tie to merge the transcripts and combine the annotation. So the answer is no, we couldn't validate most of the very long ones. And for the integration of the single cell, in fact, it's still ongoing the discussion on how to integrate it. I don't have a clear answer on what's the best way to integrate it, but we are trying to put the effort to do with the BovReg data. But personally, I don't have a clear answer on how is the best way to integrate with the single cell for the 8 issues that we did within the BovReg.
Hello, James Coltis, Iowa State. 2 questions for you. The first one, when you talked about the extra transcripts that you found in the neonate, what ages roughly were those?
You mean the
Neonatal
the
levels.
it's 2 weeks old.
Okay. Did you also have fetal samples?
No.
Okay.
On the BovReg, we don't have fetal samples, and in fact, it was one of the discussions in that project, Gene Switch project final conference, the importance of having very early development stages where we have plenty of biological process already defined, and we don't have in BovReg. Our first try was, we want to have diversity, 2 weeks old and sexually mature, but we need to look at fetuses we don't have in BovReg.
And I mentioned because the US FANG project has those fetal time points, and there are a number of new transcripts identified there. We didn't identify as many transcripts as you did in the study that you showed.
You also use a tRNA?
No, it's mRNA. Second question was about looking at the overall catalog of transcripts. Just curious, how many unspliced versions of transcripts do you identify? And do any of the technologies differ for the amount of unspliced transcripts that you identify?
I didn't quantify yet how many, but we indeed identify, especially when you combine tRNA with mRNA, because you have plenty of nascent transcripts on the tRNA that were not represented on the mRNA. So we could see that we have plenty of those on the annotation, but we didn't quantify yet how many in terms of splicing transcripts or unspliced transcripts we have. But we indeed have, and it's way more when you use tRNA. So we need to put a flag when you use tRNA to do the transcriptome atlas. You will have plenty of nested transcripts, unspliced transcripts that need to take into account on the final annotation. But we didn't quantify yet.
Are those unspliced transcripts junk, or are they useful for something? I have no idea. Just
yeah.
Curious.
Well, I think it's something that you can use to study and move. It's trying to understand the splicing mechanism and how it works on the different introns and velocity and so on. But it's also an open question for the community if they will be useful, really useful or not. I don't have a clear answer to.